Hi everyone, welcome to my channel Pure Biology, and today I come with another video, that is the termination process of the translation. Okay, and in my previous videos I have already explained the process related to the translation, so you just check out that all, and this is the last process of the translation. So here another thing also there, but this is another thing how the proofreading is occurring in the translation process. This is the another thing, but the process of the translation, that is the three including the three parts, that initiation, elongation and termination, and this is the last part. Okay, so what is saying that termination, one of the three termination codon, that is UAG, UAA and UGA is reached, then protein synthesis ends. Okay, if one of three codons there in the P site or an A site, so generally in the A site, so in that situation the protein synthesis becomes stopped. And the three codons, the name of the three codons are, UAG is known as the amber codon, UAA is known as the ochre codon, and UGA is known as the opal codon. So these three codons are very important, that creates the termination process of the translation. Okay, and obviously the factors that are called the releasing factors that are involved in this process. So first of all, what is saying that the A site is entered by a protein release factor. You can see here, this is the rough diagram. In the A site, the protein releasing factor, or you can say the codon, that is known as the stop codon, UAG, it enters at the A site. Okay. Second one is that RF1, this is the releasing factor, RF1 and RF2, that after recognizing the termination codon, activate ribosome to hydrolyze the bond between tRNA and the polypeptide. So what is happening here, when the releasing factor, they can recognize that stop codon already enters to the A site. At that situation, they try to hydrolyze the bond already formed between the tRNA and the polypeptide chain. Okay. Third point is that polypeptide is released through the glycine, that means GGG motif, which acts as the peptide anticodon. So this is released. You just learn it that polypeptide is released as the peptide anticodon due to the peptide, due to the attachment of the stop codon, it released as the peptide anticodon. Okay. Here this motif actually acts as the peptide anticodon. Next one, that RF3 and RF2. RF3 that stimulates to release the RF1 and RF2 from ribosome after termination. That means RF3, another releasing factor, just when the termination occurring, at that situation, or termination already have done, in that situation the releasing of RF1 and RF2 is occurred by RF3. Okay, RF3 stimulates to release the RF1 and RF2. Next one is that here one energy is required, and this energy is driven by or it is coming from the hydrolysis of the GDP. And in case of eukaryotes, there are only one factor, that is the ERF1. It is very important because it can help to recognize the three of the stop codons. Okay. And here ERF1 is very important, but ERF3 is also present, but what is the function of the ERF3, it is not known. Next one is that the additional protein ribosome recycling factor, it is very important. This is called the RRA factor. Its function is what? It helps to dissociate the ribosomal subunit. That means these RF1 and RF2, its function is recognize the termination codon. Okay, but RRF, its function is to release the ribosomal subunit. Okay, that means already large subunit and the small subunit, they join each other and form a complex. So first when the stop codons are recognized by the releasing factor, in that situation this RRF factor this try to break or the splits this ribosomal complex. Okay, and here this RRF acts togetherly with the EFG. Okay, here not RRF is the only one that is responsible for the releasing of the ribosomal subunit. Another EFG is a very important. This is the elongation factor. This is also very important for doing this. And this RRF probably enters the P or A site and unlock the ribosome. Okay, it can enter maybe A site or P site, and then it tries to unlock the ribosomal subunit. And dissociation requires also energy. And IF3 is the initiation factor. This initiation factor also needed here. Why? Because it prevents the subunits from attaching together again. If the large subunit and the small subunit they become released from each other, then there is a chance to bind with each other or try to make a complex, and this is prevented by the IF3, initiation factor 3. And here one thing is important, that is the dissociation requires energy. This is getting from the GTP, but here also elongation factor is important that helps to release this release the energy by hydrolyzing this GTP. Okay, so this is the termination process of the translation, and I think you can clearly learn from here. And if this is helpful to you, obviously you can share this and subscribe this channel for the growth of this channel. Okay, so thank you.